Welcome to this lecture on cell and molecular biology. Today, we are going to explore one of the most elegant and crucial processes in all of biology, semi-conservative DNA replication. We will understand exactly how your cells copy their genetic instruction manual with near perfect accuracy every single time they divide. To start, imagine you have a precious, ancient recipe book from your grandmother. If you want to give a perfect copy to your two children, you do not just photocopy it. Imagine the book is magical and splits perfectly down the binding. You give half to each child, and a magical scribe writes in the missing pages. Both children get a complete book that is exactly half old paper and half brand new paper. This intuitive idea is exactly how your cells copy their DNA. Now that we have an intuitive picture, let us look at the formal biological mechanism. A DNA molecule is a double helix, resembling a twisted spiral staircase. During replication, specialized proteins unwind and pull apart these two original or parental strands. Each separated strand then acts as a strict template or mold for building a new complementary strand. As a result, every newly formed double-stranded DNA molecule consists of exactly one old parental strand and one newly synthesized daughter strand. This template mechanism works so perfectly because of the strict base pairing rules of DNA. Adenine always pairs with thymine, and cytosine always pairs with guanine. Because these rules are absolute, the newly built strand is guaranteed to be a perfect match to the old one. The cell does not have to guess which building block comes next. The original strand dictates the sequence exactly. This ensures the genetic code is preserved across generations, allowing life to function and thrive without losing its core instructions. You might wonder why the cell uses this specific half-and-half method, instead of just building a completely new DNA molecule from scratch. The answer comes down to extreme accuracy and genetic fidelity, which are absolutely critical for your survival. By keeping one original strand in every new DNA molecule, the cell has a built-in proofreading mechanism. If a mistake is made, the repair machinery checks the old strand to fix it. Without this, errors would accumulate rapidly, leading to devastating mutations, cancers, and cell death. To mathematically prove that DNA replicates this way, scientists Matthew Meselson and Franklin Stahl designed a brilliant experiment. They grew bacteria in an environment with heavy nitrogen, so all the bacterial DNA became heavy. Then they moved these bacteria into an environment with light nitrogen and allowed them to divide. By tracking the weight of the DNA over several generations, they could calculate the exact fractions of old versus new DNA. Let us look at the mathematics behind their groundbreaking findings. This brings us to the mathematical representation of the experiment. Because one cell divides into two, the number of DNA molecules doubles every generation. We represent this exponential growth with the formula Tn equals T0 times 2 to the power of n. Here, Tn is the total number of DNA molecules after n generations. T0 is the initial number of DNA molecules we started with, and n is the number of replication cycles. As n increases, the total amount of DNA grows exponentially, doubling with every single step. Before we move on, let us see this with a concrete example. Imagine a scientist grows a single bacterium in a heavy nitrogen environment, then moves it to light nitrogen for exactly 2 generations. We are given an initial molecule count of 1, and the number of generations is 2. We plug these values into our formula. 1 times 2 squared gives us 4. This makes perfect physical sense because one cell divides into 2, and those 2 cells divide into 4. The population has successfully doubled twice. Now that we know the total population, we need to find out how much of that DNA is hybrid. A hybrid molecule contains one old heavy strand and one new light strand. The formula for the hybrid fraction is F sub h equals 2 divided by 2 to the power of n. The numerator is always 2 because the original double helix only had 2 heavy strands to give. The denominator is the total number of DNA molecules. As the generations increase, the denominator gets larger, meaning the fraction of hybrid DNA rapidly decreases. Let us apply this hybrid formula to our previous example of 2 generations. We plug in n equals 2, giving us 2 divided by 4, which is 0.5. This means 50% of the molecules are hybrid. To find the completely light DNA molecules, we subtract the 2 hybrid molecules from the 4 total molecules. This leaves us with exactly 2 completely light molecules. Physically, this proves the semi-conservative model, as the original heavy strands are perfectly conserved but separated into 2 different molecules. Let us take the exact same experiment further to see what happens over a longer period. If the bacteria continue to divide for a total of 4 generations, we want to know how the original heavy strands are diluted. We follow the exact same logical approach. Our generation number is now 4. We calculate 2 raised to the power of 4, which is 16. So there are now 16 total DNA molecules in our bacterial population. The population is growing very quickly due to exponential growth. Now we determine the percentage of the population that is entirely light. We know there will only ever be 2 hybrid molecules because we started with exactly 2 heavy strands. We subtract these 2 hybrids from the 16 total molecules to get 14 completely light molecules. Dividing 14 by 16 gives us a light fraction of 0.875. Physically, this beautifully illustrates that after 4 generations, 87.5% of the DNA is completely light, while the original parental strands are conserved forever. Now that we have calculated the fractions, let us visualize the actual results of the Mieselson and Stahl experiment. On the screen, you see 3 test tubes. In generation 0, all the DNA is heavy, forming a single band at the bottom. After 1 generation, the DNA is entirely hybrid, forming a single band in the middle. By generation 2, we see exactly what we calculated, 2 distinct bands. Half the DNA remains in the hybrid middle band, and the other half forms a new light band at the top. This visual perfectly confirms the semi-conservative mechanism. We have reached the end of our topic. Let us summarize the key takeaways. First, DNA replication is semi-conservative, meaning every new DNA molecule consists of exactly 1 old parental strand and 1 newly synthesized daughter strand. Second, this built-in template system is crucial because it prevents catastrophic genetic errors and allows life to faithfully pass on its instructions. Finally, the famous Mieselson-Stahl experiment proved this by tracking heavy and light DNA over multiple generations. You now have a solid understanding of how your genetic code is preserved.